this normal fibroblast. So mouse fibroblasts. The population doubling levels were significantly increased, and the population doubling times were significantly decreased. It means they have, they grow faster. Yeah, that is the population doubling time quantifies the proliferation speed. Also, what have we done? We had mice, as we call them, naive, so not immunologically challenged. We have sacrificed them, we have harvested splenocytes, and then we put them under the stimulation of chemical triggers like bacterial lipopolysaccharide and concanavalin A, and these will stimulate the growth of the cells. And of course, in a specific medium, and this medium was deionized water and deuterium depleted water. What have we observed? That the groups that have received concanavalin A and deuterium depleted water were growing very fast compared to the control. The control here is this violet color. And the LPS also was increased. So the group that received lipopolysaccharide increased the concentration or the proliferation of the native splenocytes. So what are the conclusions of this experiment? Cell proliferation is significantly stimulated. Effect observed both in normal cells and in neoplastic cells. Growth of and metabolism of splenocytes was stimulated in both groups, but in case of the LPS was not statistically significant. However, using concanavalin A as proliferation stimulus for the native splenocytes, we have observed a significant stimulation, especially in high doses. Now, we have thought, what might produce this? And we have thought exactly the same idea of Dr. Shomnes here. It has to do something with proton transport in the membrane. So what have we did? We put a specific inhibitor that we call amiloride, that we know is amiloride, inhibitor of sodium proton pump. In the growth media, we have observed no significant changes. So a single cell type had growth and proliferation rates similar between the two, but not statistically significant. However, I've read somewhere that it might be predominance of what we call amiloride resistance NHE3 channels. So it means that it's useless, the substance that we tried. However, in what we have studied is the probability that the antiport, proton sodium antiport, is not involved in those changes which might increase the alkalinity of the cells. We tried to do another blocker, a blocker of potassium hydrogen blocker, which is called lansoprazole and is usually used for treating ulcer because it also blocks the proton pump in the stomach and reduces the acid concentration in the gastric juice. So we use that substance, and what we have observed that it practically reduced completely the difference between the control and the deuterium depleted water cultures. So there is no difference anymore between those two. It means it has completely abolished the effect of deuterium depletion. How does it do it? Probably suggests an involvement of the proton potassium antiport in the mechanism that stimulate deuterium-induced cell proliferation. We don't know how it's done because it had to be continued, but we did not have the resources anymore. A very good idea would be to try to test using a patch clamp experiment to see if the current flow from one side to another is impaired in any way. But patch clamp experiments are quite expensive and they need a lot of expertise and machines, so we were not able to perform them. So, what are the conclusions? Deuterium depleted water is an efficient modality of reducing concentration of deuterium in animals subjected to prolonged treatment. Decreased to approximately 90 parts per million, and no prolongation was able to further decrease it. The long-time treatment significantly protects animal against it. However, it has to be given before the irradiation. The association with the most powerful chemical radioprotector did not significantly increase the effects of the diet. The histological aspect suggests protective effects against radiation-induced cellular depletion. The investigation showed that antioxidant is present but not strong enough. Most important action was the stimulation of non-specific immunologic parameters. Proliferation was significantly stimulated, and the antiport does not seem to be involved. However, the ATPase might be involved in these mechanisms. We have made a few other experiments, kind of a hobby. And what have we observed? Unsystematic data using biomass growth evaluation for Elodea canadensis. Elodea canadensis is a plant which we use in aquariums. And water sails from the Pomacea group have shown statistically significant mass increase. Ouch. Something? Mistake. Statistically significant mass increase while aquarium water was reconstituted with deuterium depleted water. So, truly, the deuterium depletion increases growth rate and speed. The exact reverse example that was demonstrated by the professor here when he showed us that heavy water stunts growth, while deuterium depleted water stimulates growth. However, what we have observed in the snails grown in deuterium depleted reconstituted media, we have observed the so-called hepatopancreatic hypertrophy, which is a phenomena characteristic to neoplastic growth types in invertebrates. And this might caution us in administration in patients with cancer. I'm not an oncologist. I never gave it to patients with cancer. It has a very good effect out of what I read and from stories. It's absolutely true that cancer treatment with deuterium depleted water seems to be real. However, it has to be made in a discriminate way, because otherwise it might, let's say, stimulate perhaps not growth of the primary tumor, but however, who knows what effect it might have on metastasis and whatever other changes induced by the cancer. So the final words after this presentation is we truly have to investigate deuterium depletion using this tool, because it's something of a very big future. There is something here, everybody feels it, and everybody would want to see it done. So let's hope this symposium is the first step towards increasing the possibilities of the research. Thank you.